Now we'll discuss components of column chromatography. So we'll discuss different components one by one. First of all, stationary phase. Stationary phase, we know, is very important in column chromatography and other types of chromatographic methods as well. Here in column chromatography, the component of a mixture which has more affinity with the stationary phase that passes slowly, while those components which have less affinity with the stationary phase, they will pass fastly through the column. A number of compounds, they're used as adsorbent in the column. These include starch, alumina, silica, many others. Adsorbents which may be selected that depends on particle size and activity of the solid. Now, activity grade is a measure of an adsorbent's attraction for solutes in the sample to be analyzed. Generally, complete anhydrous adsorbents, they have the highest activity grading. Now, mobile phase. As the name indicates, it passes through the column. That's why it is named as mobile phase. And the components of a mixture which are more soluble in mobile phase, they pass through the column fastly, while those components which are less soluble in mobile phase, they pass slowly. It is very important to introduce the mixture into the column for the separation. Its choice depends on polarity for the sample and also for the stationary phase. We have already discussed. It is the best strategy to start elution with a solvent of lower polarity for elution of the components that are weakly adsorbed to the stationary phase. These mobile phase, or the solvents which are used, they are also known as developers and as elutants as well. Now, column dimensions of the column are very important for efficient and effective separation. And this column consists of a glass tube with the bottom packed with glass wool or cotton wool or some other material, and it is filled with some stationary phase. After packing, a paper disc is kept on the top so that the adsorbent layer is not disturbed when a sample is introduced in the column. Glass which is used here, that should be neutral so that there is no effect of the solvent, acid or alkali on the process of separation. Here one can see different components of column chromatography. Column is visible. In the column we have stationary phase which is packed in it. We have mobile phase which passes through the column. On the top, where one can see input, here the sample is loaded and the sample passes to the column. At the bottom there is cotton plug, and then there is output, and through this output this elution takes place. So different components of a mixture, they are separated and they pass out of the column at different rates and at different times. Now, sample injection. A graduated pipette is filled up with the sample mixture, and this is introduced by touching the top of the adsorbent layer, having a filter paper, so that the column is not, the stationary phase is not disturbed. And then fraction collector. We have discussed that when this separation takes place, the component of mixture, they are eluted through the column at different times. So these are collected. So fractions which elute from the column, they are separated in different tubes and at different times. And fractions, they can also be collected by means of fraction collector. And when this process takes place, so we get fractions, and these fractions then they are analyzed further for different types of analysis.